hopefully everyone's tummy is starting to rumble a little bit. We have one more talk before lunch. And that's Jeremy Snell from the College of Pharmacy. And I'm not going to try to say this title fast. So, thank you.
Microphone's not? There's a microphone. You can also talk here. Okay. All right, can everyone hear me?
Yeah.
Okay, great. Okay, this works. Okay. Well, good afternoon, or I guess it's technically still morning, so, well, hello everyone. So I'm Jeremy Snell from Dr. Wondrak's laboratory, and today I'll be discussing with you environmental chlorination stress and skin health, the chlorination agents hypochlorous acid and TCIC trichloroisocyanuric acid display opposing effects on solar UV-driven inflammatory dysregulation and skin carcinogenesis. All right, so the conceptual framework that underlies this talk is the skin exposome. Okay, so as we can all imagine, the skin is among the most environmentally relevant organ systems that we have. It is the, if you conceive of it as far as exposed surface area to the outside world, especially if you look at invaginations of the skin, it's not exactly, the skin is not flat, right? We might think as an approximation that it's flat, but if you actually look, just like villi and the gut have more surface area. And so, as you can imagine, there's microbiome, there's the endogenous microbial host-associated microbes that are beneficial, but moreover, we also have microbial infections, so those are pathogenic microbes. We have allergen contact, physical traumas, solar UV radiation, pollutants, xenobiotics, tobacco smoke, drugs, pharmaceuticals, personal care products, disinfectants, and that's the part of the talk today, but also dietary and lifestyle factors. And why does this matter? Well, all these exogenous and endogenous factors interact in a very complicated, second, third, fourth order, multifactorial milieu, this whole mix here on the skin that then has all these effects on human health, skin outcomes such as skin barrier dysfunction, infection, immune dysregulation, inflammation, impaired wound healing, photo damage, photoaging, cancer, etc. And so we'll get back to, as you see on the upper right here, we're looking at disinfectants. And particularly, we're not just talking about any disinfectant, we're talking about chlorine-based disinfectants. And so someone might say, well, wait, what are you talking about, bleach? Is it really that interesting? Well, the thing is, if you add up disinfection of water supply, drinking water, you add up recreational exposures and also household and industrial use of chlorine-based disinfectants, 25% of the human population is exposed chronically to these chlorine-based disinfectants. It's a low-level chronic exposure, but it's still there. And so the Wondrak lab, we're photobiology, we're looking at skin cancer, inflammation. This is our area. So a couple years back, we were thinking, what would be an interesting, we're thinking Arizona, arid environment, UV, sun. What's an interesting co-exposure? Well, swimming pool. People go to the swimming pool during summer, and they're probably getting a lot of, UV exposure coincident with chlorine being a highly oxidative species. So we first thought, okay, let's go ahead and look at this. Let's look at this idea. So chlorination stress and health outcomes. So I'll just go over this briefly. But we have, here we conceive of primary chlorination stress inducers, top left in A. So these are primary disinfectants, hypochlorous acid or chloramines or chlorinated isocyanurates, which we'll get back to later. We also have disinfection byproducts, particularly here we're just concerned with chlorination byproducts. But moreover, and disinfection byproducts is a whole other topic, but let me just say that government only really concerned itself in regulating 11 of these disinfection byproducts, but there's many hundreds or thousands that we know of. Now, the idea is that those 11 are a good proxy for the mass aggregate exposure of the human being to these disinfection byproducts, but that's a whole other area that needs to be looked at, and there's a lot of research ongoing in that area. Okay, so disinfection byproducts, that's a secondary source of chlorination stress, but moreover, we also have all of our lotions, all of our skin cosmetics, etc. Imagine if you're in a swimming pool scenario and you have chlorine, right, that reacts with drugs or organic chemicals, what have you. Now you're having chlorinated byproducts, PPCPs we call them, right? Chlorinated byproducts. And as you see, chemical entity 14 and 15, that's the sunscreen, common organic sunscreen, that you have this dichlorinated species that's been described in the literature, but nobody knows what it does to human skin, or moreover, a lot of these organic sunscreens were approved decades ago, and they actually have systemic exposure. So imagine you have a chlorinated sunscreen byproduct that could technically be floating around your systemic circulation. Nobody knows what it does. Anyway, okay, so then chlorination stress pathways, we have There's been some stuff described in the literature, so there is some molecular biology out there that's been established. Moreover, there's lung, GI tract, liver, kidney, bladder, skin. These are all the tissues of interest that have more. Most of this is described in the context of disinfection byproducts, but here we can concern ourselves. These are main tissues that are going to be affected by chlorination stress. And then paradoxically, or perhaps counterintuitively, you'll see some of our data that presents that hypochlorous acid also has therapeutic opportunities. And it's obvious in dermatological contexts, wound cleansing, dental situations, etc. We actually show that here, in our first data I'm going to show you, it actually has anti-inflammatory and anti-tumorigenic properties as well. Okay, so chlorination stress and solar UV co-exposure. So this is our SKH1 mouse model. This is a well-established model for UV inflammatory signaling, etc. Okay, so I'll just get into this really quickly, but all I want to basically draw everyone's attention to here is in panel D. So we just did a gene expression analysis. This is an oxidative stress qPCR array. And as you can see, inducible nitric oxide synthase. So let me just describe. So column 1, HOCl column. As you can see, there's a few genes that are upregulated here. For instance, glutathione synthetase, 45.8-fold. We have a few others, so on. Really, just look at the central column. You see that UV, for instance, nitric oxide synthase. And this is basically blood vessel dilation and redness and skin, so on. Inflammatory, right? UV, 486.6 upfold, upregulated, right? We have Hypochlorous, another well-known inflammatory mediating gene product. So as you can see, UV has a redox inflammatory sun responsive or UV responsive gene expression regime here. But as you can see, the co-exposure scenario, hypochlorous acid, that previously we saw 486.6 upfold upregulated for nitric oxide synthase. Here it's completely ablated. And moreover, for instance, all these others in the middle, all these other gene products that are redox or inflammatory, they're also ablated. So it was kind of counterintuitive. We thought, surely oxidative species plus UV, it's going to be inflammatory. No, it actually wasn't. On the gene expression basis, we found it was anti-inflammatory. So we have another tool, which is our AP1 luciferase mouse. Okay, so these are basically genetically engineered mice that have luciferase under the control of the AP1 promoter. AP1, it's the central regulator of UV inflammation, just transcription, a family of transcription factors, mediates inflammation, but also epidermal differentiation, and also critically involved in carcinogenesis as well. Okay, so as you see, we have 4 mice here that are representative of these 4 groups. All the way to the left, we have our control, then we have our UV alone, then we have our HOCl, and then we have our co-exposure. By the way, I forgot to mention this earlier, but we had done previous data. We took the upper environmentally relevant range of HOCl that's considered safe, 100 micromolar, or 5 part per million. And so for all the mouse data you're seeing here, these are environmentally relevant human possible dosages that could be occurring on your skin in a swimming pool context. Okay, so control. And so here what you're looking at is the signal, the bioluminescence signal. So control, we don't see any bioluminescence, which means that AP1 is not active. There's no UV-induced inflammation. As you can see, the second column, there's quite intense, there's a statistically significantly upregulated signal of the UV group. Then HOCl alone doesn't do anything. And then the co-exposure is statistically significantly different. It's suppressed. So once again, we see also on a bioluminescent AP1 luciferase model that it also jives well with our gene expression data. And just to double check this, we also did some single PCRs of some downstream genes of AP1. As you can see, the UV upregulates these genes, and then the co-exposure suppresses that signal once more. So we have gene expression and AP1 luciferase data that both agree with each other, that hypochlorous acid substantially downregulates or suppresses UV inflammation. So then we go into our next model. This is our photocarcinogenesis long-term high-risk mouse skin model. So they're the same mice. Okay, so these are SKH1. These are hairless mice. I don't know if I mentioned that before. Hairless mice because obviously if you have hair or differing amounts of melanin, etc. So these mice are this is the closest established proxy of photoinflammatory, UV, carcinogenesis, etc. that you'd see in a human being in a mouse model. So this is well established and well known. And so we have a 112-day regimen where the mice are irradiated 3 non-consecutive days. So like a Monday, Wednesday, Friday, they're receiving a mild sunburn basically. So as you can imagine, a mouse lifetime, this is an approximation of a chronic lifetime of low-grade UV exposure over time. Then we give them a gap phase. And as you can see in panel C, this is a tumor growth, tumor burden data. And so as you see, after the gap phase, we basically irradiate them for 4 months approximately. We give them a gap phase, and then they start developing these papillomas, these tumors. And then in that phase, the post-UV phase, right, as you see, we have this exponential increase in these tumors. In that phase, we apply, once again, the 100 micromolar HOCl to the skin topically, and we statistically significantly reduce tumor burden in these mice. So again, basically that was our first paper. And then this does, I'll just speed it up here, but also on the tissue-based IHC data also agrees with everything we saw previously. So on a gene expression level, imaging level, tumorigenic readout, and also on our protein-based assays, they all say hypochlorous acid is anti-inflammatory following UV, or suppressing UV, rather. But then we thought, okay, well, that's not technically the case in the swimming pool, because the thing is with hypochlorous acid, it's actually photolabile. Depending on the pH of the aqueous chemistry, basically, depending on the pH, it's going to be degraded by sunlight. And so in swimming pools, you actually have photostabilizing agent, isocyanuric acid. So here we said, okay, let's look at trichloroisocyanuric acid, which we would see in a swimming pool. We did the same exact exposure, same exact everything. And as you can see, there's some common household products that have TCIC as the active ingredient. And so once again, as you see before, TCIC is compound 5 here, so that's one of our chlorination stress inducers we talked about before. And we repeated the same experiment, so I won't go too much into detail, but as you can see, it's the exact opposite. Whereas previously we saw that the chlorine species alone, the chlorine species co-exposed with the UV statistically significantly downregulated that UV-induced inflammation. Here we see the opposite. It potentiated it. So I think this is quite a striking figure, and I think it's just meditating on this is really quite exciting. It's striking. And so once again, on a gene expression basis, we see the same thing, that it actually potentiates. We're in the same qPCR panel, and we also see, okay, it is enhancing the inflammation. It's the opposite of what we saw before. And again, single PCRs are in accordance with this, and our IHC is also in accordance with this. And so here's just a highlight. Somebody else published it. We attracted some attention. And so this is a highlights summary page that kind of puts all of it into context. And so we have some interesting stuff in the works currently. First thing is investigation of the impacts of chronic chlorination stress on gastrointestinal epithelial tissue, because the majority of us, unless we're constantly swimming, not many of us are constantly swimming, for the majority of us, the more relevant exposure is going to be gastrointestinal, aka drinking water. Okay, and then beyond that, we also found, we're the first to describe this, we actually found a novel, we're in development phase, we're getting there, but we're pretty confident we found a novel skin biomarker of chlorination stress. And that's both on a, in vitro, and we have some ex vivo data as well on that. That's where we are currently. And so, thank you very much. I'll take any questions.
Thank you. We have time for a couple of questions. John?
Why do they have opposite effects?
Oh, okay. So basically the whole thing about isocyanuric acid is that, so as we described earlier, that hypochlorous acid alone is photolabile. It absorbs UVB. Isocyanuric acid, or isocyanurates, they're far UVC, so they're like low 210, 220-ish nanometer, depending on ketoenol tautomerism and so on. And so what it is that you have these, they're chloraminated. The isocyanurate has these nitrogens that can form chloramines, and then via reversible hydrolysis, you can yield equivalents of hypochlorous acid. And so what I think is going on is, so basically the chlorinated cyanurate can pick up hypochlorous acid or release it, depending on chlorine demand and so on in the aqueous condition. And so what I think is going on is that in our first case, you apply HOCl, it's highly reactive. And so it's reacting with amino acids or lipids or what have you, forming all these intermediates that are longer lived, more diffusible. For instance, there's like taurine chloramines, well described in neutrophils. So I think in the first case, HOCl that's not in buffered by that organic precursor. The first case, it reacts and it does its thing and it's done. But by the chlorinated cyanurate, via that reversible picking up and giving of hypochlorous acid, UV, actually, hypochlorous acid, as we know, it splits and yields all kinds of radicals, and there's all kinds of stuff going on there. So I think what's going on is by prolonging that lifetime, by recycling hypochlorous acid over and over, and obviously the aqueous chemistry is different than topical. Obviously, they're very different environments. But I think we could be enhancing the statistical probability of a UV photon incident on an equivalent of hypochlorous acid or another chlorine species intermediate, and then yielding interesting radical species that are then causing the inflammation. So I think that's what's going on, is that the first case, HOCl, it reacts and it's done, whereas the second case, because of that recycling effect, we're having more and more and more radicals being spit out. That's my only rationale at this point. And nobody knows. We're the first ones to even figure this out. Nobody even knew this until us, so it's cool.
New discoveries are always great. Any other questions for our speaker before we break for lunch? Okay, so we will reconnoiter at 1 o'clock for the second part of our water and